right, everyone, welcome back. I hope you had a very nice break. It is my pleasure to introduce the next lecture, which will be held by Johanna Beyer. And just in case you missed the introduction yesterday, where she already introduced herself, I will repeat a bit of the information again. So Johanna Beyer is a research associate and lecturer at the Visual Computing Lab at the Harvard University in the United States. Before joining Harvard, she was a postdoc at Geometric Modeling and Scientific Visualization Center at KAUST. She received her PhD in computer science at the University of Technology in Vienna, Austria in 2009. Her research focuses on GPU-based volume rendering techniques for large-scale neuroscience and medical data, with an emphasis on visualization of large and multimodal volumes. Today, her lecture will be about the visualization of neuroscientific and connectomics data. Johanna, it's a pleasure to hear from you. Please go ahead.
Great, thank you very much. Can you guys all see my slide and hear me? I assume yes. Wonderful. Cool. So thanks for the nice introduction, which means I can basically skip this next slide, which gives me more time on the actual connectomics part. I just added some nice pictures of my progression through the world from Austria to KAUST to Harvard. And also a shameless plug at the Visual Computing Group. We are also hiring right now. So if you're looking for a postdoc position, contact us. Generally, our research is not just visualization, but also computer vision and machine learning. And we've done projects in the areas of neuroscience, but also in genomics, pharmacology, and cell biology. So it's kind of a widespread team. I'll focus, and especially in my talk today, on the neuroscience part.
I'm sorry, Johanna, we cannot see you, unfortunately.
You cannot see me?
Your video somehow.
Interesting. Let me try to fix this. Now you can see me?
Yes. Thank you.
All right, let's try again. All right. Yes, so let's continue with the actual talk about neuroscience and connectomics. So first of all, what is neuroscience? And I would say in a very general sense, any of the sciences that deal with the function and the structure of the nervous system and the brain. And generally, that also means that neuroscientists can work at completely different imaging scales. And yesterday, we've already heard a little bit about fiber tracts and visualization of that. So that's what I would consider like the macro level neuroscience. And then there's also meso or micro and nano level. So in the meso level, you can see in the picture in the middle here, individual cells all highlighted in different colors. And on the right, there's an electron microscopy data set with really high resolution structures where you can see substructures in a cell. So in this talk, I'll mainly focus on the nanoscale brain connectivity. So it's really about electron microscopy and getting the highest resolution possible, which allows you to image like individual synapses and substructures in a cell. All right, so that means I'll focus a lot on connectomics. And first, maybe a connectome is basically a map of the neural connections in the brain. And the main goal of connectomics is to reconstruct this full wiring diagram of the brain. And that is a huge effort because the human brain consists of around 100 billion neurons that are interconnected with thousands of connections to neighboring neurons between one neuron and the other. And in this picture, you can actually see a so-called rainbow data set where each neuron is highlighted in a different color using fluorescent proteins. And this was imaged with confocal microscopy. So that's a resolution lower than the actual electron microscopy data, where you can see individual cells in much higher resolution. All right, so talking a little bit more about connectomics data, what is connectomics data? What do we usually deal with? So first of all, we deal with large image stacks. So either light microscopy or most commonly electron microscopy, where we get a stack of 2D slices and then we form the 3D volume and we can do stuff like volume rendering on it. In addition to that, we get segmentation image stacks, which are usually dense segmentations where basically for every pixel in an image, we know which structure it belongs to. In addition to that, we often have connectivity data, so knowing which structures connect and where. So between neurons, there are synapses, so we have either the location of synapses and usually also the IDs of the two structures they connect. And then there's a bunch of metadata that we might have where neuroscientists either manually labeled stuff or kind of automatically extracted additional information. So we have a bunch of different data sets that usually tend to be very large and very complex. So here I'm actually going to start a video. So this is just a subset of a slice of an EM data set. So what neuroscientists do is that they collect this stack of slices to form this 3D volume that you can see, and then they do the segmentation. And that means for each slice of the data, they kind of define which pixels belong to a certain structure, such as this neuron that you can see. And they do that for all the slices. And then that means you can reconstruct this labeled neuron in 3D based on all the segmented slices. So here you can actually see two structures that have been segmented, and now we zoom back in on the EM slice where they form a synapse, and if you look closely, you can see that here they form a synapse. All right, so if we continue from here, if I can move on to the next slice. All right, yeah, so here I've highlighted the synapse between an axon and a dendrite. And neuroscientists are very good at just looking at the EM data and seeing that, but it's kind of tricky if you're not trained on this. And now if you do this reconstruction that you just saw in the video, not just for two neurites, but for the entire volume, you get something like this. And you can already see now we have a cube of data that is so dense and so complex that we don't see anything anymore. So if you really want to analyze connectivity, it's kind of difficult if you just look at the 3D data here. So that's why we really need kind of clever and interactive visualizations that allow filtering and kind of analyzing the data more than just rendering it in 3D. All right, so coming back to the electron microscopy volumes that we have. I already mentioned that they tend to be very large and very high resolution. So we often have a pixel resolution of 3 to 5 nanometers. It's usually an anisotropic volume, meaning that the slice thickness is much thicker than the resolution within a slice. But if you use that resolution, even like a cubic millimeter block of tissue will result in 800 terabyte of data. So that's a lot of data to deal with. And another problem is also that the acquisition of that actually kind of takes a while, right? Because it's so much data, you have to deal with tissue, with artifacts, and so on. So even if you had like a high scanning rate, such as 40 megapixels per second, if you want to scan an 800 teravoxel data set, it will take you about 8 months. All right, so here's the entire connectomics workflow, I would say, that neuroscientists follow to acquisition and kind of analyze their data. And they usually start with tissues from either a mouse brain or a rat brain, and then they use a microtome to cut it into very thin slices. And of course, this figure here is just a knife, but they don't just use like a manual knife. They have like these big machines that are able to cut tissue in extremely thin slices. And then they use electron microscopy to image it in high resolution. And then the next steps are registration, forming the 3D volume, doing the segmentation, and then in the final step, we come in and do like visualization and visual analysis. And today I want to focus on those last two parts and how we can use visualization, not just in the last step, but also already in the segmentation step. All right, so for visualization for segmentation, I would say that the goal is, on the one hand, to verify and correct segmentation results. So being able to quickly look at the segmentation of this large data set and or going in and kind of correcting it, and also tracking how the segmentation is progressing. If you have several people, for example, doing proofreading and adjusting the segmentation, being able to check that quickly. And I want to talk about three approaches today. One is really just visualizing the segmentation result. The next one is visual proofreading, and then visualizing the segmentation provenance or the progress. here's an example of how you can visualize a segmentation result. And I really don't want to go in too much detail here, but you see a very big slice view where each segmented structure has a different color. And here it's actually overlaid with the original EM data. So you can still see like some color fluctuations to see what is inside a cell. Alternative, you could also only show the segmentation mask, which would basically result in a flat color where you don't see any internal structures of a segmented neuron. The next step after the automatic segmentation would be visual proofreading. And here the idea is that even though automatic segmentations algorithms are improving all of the time, they're still not perfect. And you will always have to go in after the automatic segmentation has completed and kind of correct small errors and mistakes. So the idea is with visual proofreading to create a tool that kind of semi-automatically, hopefully, guides users to where errors are and lets them correct those errors quickly and easily to be able to go through the entire volume. So if you look at these image slices here and focus in on this part, and let's zoom in a little bit here, you can see there's this one structure in the middle that kind of looks a little bit off, that probably has a mistake. So you can see that there are actually, those are two structures that should have been separated, and then there are several small structures that are right now labeled as individual objects that should be merged. So if you use visual proofreading and you go in to correct this, what you want is an end result like that, where you kind of have corrected that segmentation. And really, you want to make this easier and faster for humans to do, because you have such big data sets, right? If you just label each pixel manually, that will take too long. So you kind of want approaches that are semi-automatic, that compute stuff in the background and kind of guide the users to the parts that need to be corrected. And here I just want to very quickly show one example called Dojo, which is a tool that is web-based for collaborative proofreading. So you can see there is the EM image slice, and in addition to the slice, they get a 3D view. They can log objects that have already been segmented that are then shown in this checkerboard pattern, basically. And the UI allows for like an easy and fast correction of the segmentation. I actually have a quick video of that. Let me start that. I'm going to skip ahead a little bit in the video. So here you can see that a user can, just by clicking on segments, merge them together and correct the segmentation like that. Or they can also draw a line, and then automatically in the background, it's computed how those structures could be split. And in addition to that, if that doesn't work, you can still manually paint and adjust the segmentation. All right, so that was just a very quick intro to this tool. Another example for visual proofreading is called targeted proofreading. That takes a little bit of a different approach, where you don't want to go through the volume slice by slice, because as I said, the volumes are so large that even proofreading such a big volume becomes unfeasible after a while. So in a targeted proofreading approach, the idea is that you focus more on connectivity related errors, and you focus on proofreading of one neuron after the other, so that you can kind of focus on the area that you're interested in, and maybe focus on the circuit that you're currently working on. And the way you can do this is you start with the segmentation, and then you can automatically compute skeletons. You can take synapses and associate them with the skeleton, automatically aggregate the synapses into cluster, so that you do all of this automatic processing in the background, basically. And that means you can guide users to areas where you think, hey, there's an error, there should be something connected, or there it seems like there are structures that are disconnected, to kind of quickly go in and correct the connectivity. Because in the And for connectomics, one of the main goals is really the wiring diagram, the connectivity. And if there is like one pixel mislabeled, usually that doesn't matter, right? What really matters is that you have the connectivity between structures correct. All right, and now I want to move on to another area of visualization for segmentation, that is visualizing the segmentation progress and focusing more on provenance. So until now, I always showed you like basically the complete segmentation result. But oftentimes it's kind of done over months where you improve a segmentation, maybe do some proofreading that will take a while and so on. So the goal in this area of research is really to track the evolution of the ongoing segmentation, so that when someone fixes errors, that you kind of can track the progress, kind of like a Git for segmentation, right? Where you can go back and say, oh, this changed, now this was adjusted and so on. And in addition to that, kind of using that provenance tracking also for task management, being able to kind of track task completion or managing a large team and letting people work on different proofreading areas. So an example here would be a tool called Neuroblocks that you can see here. And basically what we do here is that we give a high-level overview of all of the available data. So you have this pixel view in the middle that shows all the segmented data, but only as like very high-level overview, but it gives you an overview of either the completion or the size of an object and so on. So let's see, I have some figures where I can walk you through the system. So basically, this is the main view where each pixel shows an overview of the current segmentation state. So you can color code it and saying sort by when was this last edited, or is how much is this already proofread in percentage and so on. In addition to that, you can click on one structure and can get details to it, seeing it in 3D or seeing the connectivity of this one dendrite that you've selected. You can also look at individual segments in more detail and see stuff like who created it, when was it last edited, and what is the history of the segmentation. And in addition to that, you can create tasks and assign them to other people. So an example would be here, segmentation task, where you can describe what needs to be done, assign it to someone and see kind of what has been done to complete this task already. So it's kind of more of a provenance tracking thing in a like month-long segmentation and proofreading project. Yeah, in addition to that, it needs to be integrated, for example, with Dojo, like the tool for proofreading, to kind of get the information from the actual proofreading data. All right, but that's all I want to say about visualization tool for segmentation or for proofreading. The rest of the talk I want to spend on the actual last step. Once we have the data that's kind of proofread to the point where it's usable and semi-correct, doing like the visual analysis on it. All right, and for the using visualization for analysis, the main goals that I would say in Connectomics is, on the one hand, just the visual exploration of the Connectomics data, right? You have such big volumes and you want to get a first impression what's in the data. But then a big part is the quantitative analysis, and in particular, the connectivity analysis. So really figuring out which structures are connected, what kind of networks do they form, are there any motifs and stuff like that. And the approaches that I want to talk about today are, on the one hand, just scalable EM volume visualization, and then going a step further, doing two visual query systems. I'll also talk about connectivity visualization and neighborhood analysis. All right, so for scalable volume rendering, you've already heard about volume rendering, right? So the idea is that you can assign optical properties like color or opacity via transfer function to kind of decide what you want to see in the volume and what not. And here's just as an example, a slab of a CT volume. So in volume rendering, you can change the transfer function and say you want to remove all the air. You want to set the skin and the brain to semi-transparent. or you want to show contrast enhanced vessels, or just assign colors. So one main problem with volume rendering for EM data is that many EM volumes are just extremely large, right? So if you have a Connectomics data set that's a terabyte in size, doing interactive volume rendering just suddenly becomes a lot more tricky than if it would just fit into GPU memory, for example. So that means that a lot of work has actually had to focus on low-level scalability issues, making sure that the visualization system can handle these large volumes, right? So not even going into like Connectomics specific details, but really first handling scalability issues of large volume data sets. And I don't want to talk about too many details here, but like some ways how you can deal with large volumes is that if you look at this, let's say this is a volume here of an EM data set, you can use techniques like bricking, right? Where you then only need to use or need to load those bricks that actually contain interesting data, or you can say you only want to load those bricks here in red that are actually visible during volume rendering. So there are a bunch of techniques that kind of help you manage the amount of data you need to load or the data you need to process, where a lot of research has been done. But let's assume we have figured this all out and we now have volume rendering that is scalable to very large data sets. The next step in Connectomics would be to really create visual exploration tools and visual query systems that allow you to explore this data in more detail, because in addition to being a large volume, we also have thousands of structures in that volume, right? So we have segmentation data that has tens of thousands individual segments in the volume. So just being able to explore this manually is a little bit too much to handle. So you need some clever tools that kind of help you analyze the data. And one way to do that would be visual query systems. Here you can see two examples where where the goal is they should be easy to use for a domain scientist, right? So that domain scientists can pose queries and say, show me those things that I'm interested in and don't show me the others, without having to manually select or deselect every one of the 10,000 segments in the volume. And one example here is Connectomics Explorer, which is a tool for domain-specific queries that is based on a query algebra. So basically what that means is that we deal with sets and we have operators and predicates on these sets, and then we build queries based on this. And to make this a little less abstract, what you can do is that you say, all right, let's take all axons in my volume and let's take this one dendrite that I selected that I'm interested in. Now show me only those axons that actually connect to this dendrite. So you have the connectivity information. Or you can build on top of this first query and say, well, now if I start with the volume again, I'm only interested in a certain region of interest. Now, what happens if I only want the intersection of the region of interest with those structures that connect to the one dendrite? So being able to kind of really dynamically create those queries to zoom in on the parts of the data set that you're interested in. So here's a video of this. So it's going very fast, but on the right is just a very abstract node link diagram. And on the left, you can see a volume view where now the user selected one structure and now they're building the visual query and saying, all right, from this red dendrite, show me everything that connects to this dendrite, and also applying a region of interest. So basically really, the example that I talked about before, interactively being able to zoom in on the parts of the volume or structures that you're interested in. All right. And then another very, very big part is connectivity analysis. So once you have the segmentation and you also know which structures are connected, a big part of the research of neuroscientists is kind of looking at this wiring diagram and figuring out, are there any patterns, are there any motifs, and so on. So there have been a bunch of tools and a bunch of research on just connectivity analysis and kind of going from the concrete spatial 3D structures to a more abstract visualization that focuses on the connectivity. And one example here would be Neuraline. So this is a tool that's kind of inspired by a subway map, where the idea is you go from the original 3D structure to a simplified 2D map, where you still see some information of the old data. So here's an example of this. On the left, you can see the original 3D rendering of a dendrite, and on the right, you can see an abstracted 2D representation of it, where basically to go from the left to the right, you first compute the skeleton. Then based on that skeleton, you compute a 2D representation where you retain like branching positions and the length of branches, but you abstract like some of the complexity and the 3D structure. And then you project the synapses onto the skeleton and cluster them if they're overlapping to like clearly show where the synapses on the structure are. And then going back, if you have this cluster of synapses, you can basically fan out this cluster and explore individual synapses. And here the idea was, we're not interested in one structure, but many structures and how they connect, right? So showing several structures and then using interactivity to kind of show which structures are connected, or are there several synapses between the same two structures that are connected, and doing that in an uncluttered, kind of easy to navigate view. All right, and I think I'm going to skip the video for time reasons. I want to go to the last final topic I want to talk about, that's neighborhood analysis. So the reason scientists are interested not just in the pure connectivity, but also in spatial neighborhoods, is that the local neighborhoods, for example, around synapses, might actually influence the connectivity or the synapse strength, like the function of the connectivity. So what's interesting to look at is not just the neuronal structures in the brain, but for example, also the non-neuronal structures such as glial cells in the brain that are like the supporting structures in the brain. And then doing proximity analysis or comparing neighborhoods and seeing how do neighborhoods differ in young mice versus old mice, for example. So here's just an example of a neighborhood around a mitochondria. The mitochondria is the red part in the middle, surrounded by a dendrite, and then purple you can see individual synapses. And it's important or interesting to scientists to know, like, where is this mitochondria? Is it close to like big synapses, or does the size of the mitochondria kind of relate to how many synapses there are, or stuff like that? And one example would be astrocyte analysis. So here, on the top right, you can see light microscopy. In the zoomed in version, you can see a rendering here on the bottom right. But the idea is you want to look at astrocyte morphology, which is an astrocyte is a type of glial cell, and they have a very complex morphology. So if you want to analyze proximity patterns, for example, if you look at the image on the bottom left, you can see just some neurites. But if you add the astrocytes to the picture, you'll get something like this, where suddenly the astrocyte is shown in red, but if you add it, you don't see anything anymore in the rendering again, right? So kind of that makes it difficult to see any proximity patterns or distribution patterns. And another example would be here on the right, in green, you can see individual glycogen granules, and kind of looking at the distribution of those glycogen granules in astrocytes, because glycogen granules deliver energy. So it might be important where those are and if there are synapses nearby, or what type of synapses are nearby. So really looking at this entire neighborhood or spatially related neighborhood is important to scientists. All right, and I'm running out of time. So the main things you should take with you is that visualization is helpful in all steps of the connectomics pipeline. And I focused on the segmentation visual analysis today, but it's really in every step, visualization can be helpful. And then the main challenge in connectomics throughout is scalability. On the one hand, that's computational scalability, for example, doing volume rendering on this terabyte-sized data set. But on the other hand, it's also visual complexity. So creating visual abstractions that scale with the amount of structures you have in your data set or with the complexity. So coming up with clever visual encodings. All right, and here's just a slide to thank my collaborators, and that's it from my side. Thank you.
Thank you very much, Johanna, for a wonderful talk. We have a lot of questions on the Discord, but in the interest of time, I will only ask you the first one, and I hope you're willing to take the rest afterwards on Discord.
Yeah, sure, I'll go to Discord after.
Yeah, Klaus Eckelt is asking, who would and why would you make and segment a new scan? If it takes multiple people and months, he assumes it needs a very good reason to create another.
Yes, good point. So one thing is that different neuroscientists are usually interested in different aspects. So they might want their data set like of one part of the brain versus another person use another part of the brain, or they might just stain it slightly different to highlight the features they're interested in. So that's one thing. I feel biologists also sometimes want to work on their own data, so being able to do their own research on their own data. And another thing is just that technology improves a lot, right? So also the electron microscopy and the staining and the slicing, all of that improves over time. So as new technology comes out, you can do different things, or you might get higher quality data with less noise, where suddenly your segmentation algorithms work a lot better. So those are some of the reasons.
All right, thank you very much for this talk.